Now we are here with Marco Prado. He is a symposium organizer and presenter. Thanks for coming, Marco.
It's my pleasure, Charlie.
So, Professor, tell us about your symposium here in this year's meeting.
Yeah. So our symposium, which brings people from all over the world as speakers, it's about novel process and mechanisms in Alzheimer's disease. So there is a mainstream current of thought in Alzheimer's disease that is driven by the pathology in the disease. The speakers of the symposium are going to talk about other mechanisms that are not part of that mainstream and discuss new discoveries, but also discuss new mechanisms that may be important for development of pathology in Alzheimer's disease. So we'll start with my talk, and my talk is going to focus in research that we've done in collaboration with some of my Brazilian colleagues in São Paulo. We're targeting a protein called the prion protein, which is the protein that causes mad cow disease and might be a receptor in Alzheimer's as well for toxicity. The second speaker is Hermona Sorek from Israel. And Hermona is a researcher that for many, many years has tried to understand how specific molecules called messenger RNAs and microRNAs, which are important for the regulation of the proteins that are present in your cell, so how they are regulated. And she finds that they are completely abnormal. This messenger RNAs and even microRNAs, they are very abnormal in Alzheimer's disease very early. Then we have a researcher from the United States, Eric Klan, and Eric has been a very important researcher to understand how neurons produce proteins, so how they translate that messenger RNA and produce proteins. Finally, we have a researcher from the UK, Lisa Saxida. She is in Cambridge, and she has made very important contributions for our understanding of these mouse models of Alzheimer's disease.
And how do you think your research will impact the future of Alzheimer's?
Yeah, so my particular research, which looks at Alzheimer's from several different points of view, it's the major impact that we see in the next few years is to look at new treatments, to look at new targets and potential new treatments for Alzheimer's. So that's what that's our aim in the next few years. Yeah. So it's really try to understand the disease better, try to understand the pathology, try to understand the chemical changes that occur in the brain with Alzheimer's disease. Model that in mice, because mice are the best models for that, and then use those models to understand the pathology, but also to test new drugs and new compounds, new chemical compounds to treat Alzheimer's disease.
Great. Thank you very much.
No problem.
Enjoy the
conversation.
conference.
Thank you.